Next, I would like to invite Marcus Lynch. Dr. Lynch is Assistant Manager of Horizon Scanning with ECRI Institute. And just after listening to what the Federation of Pharmaceutical Manufacturers and Associates has to tell us, Marcus is going to tell us what's in the pipeline and what is coming. Marcus, please.
Thanks, Yarin. Hello, everyone. My name is Marcus Lynch, and I'm the Assistant Manager of Horizon Scanning at ECRI. Horizon Scanning is a systematic process of identifying emerging healthcare technologies with disruptive potential and various endpoints of interest. So this is a very stimulating time in infectious diseases and vaccine development. And I apologize to my international friends that my focus will be largely US-centric just to manage the scope of this presentation. COVID vaccines are becoming available in unprecedented timeframes due to the Herculean work of Operation Warp Speed, which is a public-private partnership designed to bring vaccines and treatments rapidly to the market to address the pandemic. In a table shown, I've highlighted 5 vaccines in late-phase development in the United States that use 3 different development platforms. So mRNA vaccines, which are shown in the gold box, are the only vaccines in the US right now that are authorized for preventing COVID. However, people in lower priority groups who might be eligible for vaccination even as soon as March might also be offered adenoviral vectored vaccines, and people becoming eligible for vaccination even later than that might also be offered recombinant protein vaccines, which are shown in the gray box. Next. So at the top of this slide is a schematic of the SARS-CoV-2 virus, which is the virus that causes COVID-19. And at the left is a depiction of mRNA vaccines consisting of the RNA genetic material that encodes for the SARS-CoV-2 spike protein. And this is encapsulated in a proprietary lipid nanoparticle layer. And this aids in delivery of the vaccine into our cells. In the middle of the slide on the bottom is a schematic of an adenovirus vector vaccine. This consists of an attenuated virus that is thought not to be able to cause disease or replicate within our bodies and cause serious complications. And so this is used to deliver the genes of interest for vaccination. So for the COVID vaccines, the vector delivers DNA that was engineered to include the SARS-CoV-2 spike protein. And this DNA is surrounded by a protein shell and the vector's own spike protein, which is engineered to allow entry into our cells at the injection site. So on the right is a schematic of a protein subunit vaccine, and this uses well-established methods similar to vaccines like a hepatitis B vaccine or an HPV vaccine, where the virus proteins are grown in culture, they're purified, and then they're used in vaccines. Next. So briefly, both mRNA and adenovirus vector vaccines, they fuse with our cells and they release their contents into the cells at the injection site. Once their respective genetic material is inside of the cells, inside of antigen-presenting cells, the vaccines then make use of our cellular machinery to produce endogenous SARS-CoV-2 spike protein. And then this spike protein is expressed on the surface of antigen-presenting cells. That spike protein is also then recognized by our immune system as a foreign protein, and an immune response is produced, which includes antibodies, helper T cells, cytotoxic T cells against the spike protein. So then the vaccine components are eventually degraded by the body. Next. So some considerations for the mRNA vaccine platform: both the Pfizer and the Mrna vaccine require 2 doses, and those could be 3 or 4 weeks apart. They don't require cell culture, and their chemical synthesis has allowed a very rapid development once the sequence was known, and it has also allowed unprecedented large-scale manufacture. Some of the disadvantages of the vaccines remain that there's still a lot that we need to understand about the long-term safety and efficacy of these vaccines. Also, the Pfizer vaccine is temperature sensitive, and there's also some handling considerations. Next. So some considerations for the adenovirus vector platforms: the AstraZeneca vaccine is offered 2 weeks apart, sometimes, or 2 doses 4 weeks apart, even sometimes up to 12 weeks apart. And the J&J vaccine is currently being evaluated as a single dose. And this vaccine might be of interest to people who are in nations where follow-up visits and record keeping are particularly challenging. Some of the advantages of adenoviral vector vaccines are that they only require standard refrigeration and standard handling. In the United States, the Ebola vaccine that's approved uses a viral vector vaccine, although it uses a slightly different vector, the VSV vector. And that vaccine has been shown to be safe and effective, and so commercial manufacturing for that vaccine has been established. Some of the disadvantages of adenoviral vector vaccines are that the actual vector has its own antigens, and those could potentially cause reactivity in people. And there's an issue regarding whether or not existing immunity to that vector could limit immune responses in the future. And so there's a question of, if we need annual vaccines year after year, would you be able to use an adenoviral vector vaccine, the same vector, like year after year with a new, with an updated spike protein sequence? So that still needs to be better understood. Or if there's a pandemic with a different type of pathogen, could you then just put it into the adenovector and it would still work? Or would our immune system clear it out very quickly? So again, these need to be better understood for this platform. So it's a novel platform, and there's still a lot of investigation going on regarding this. Next. So some considerations for the protein subunit platform. Also on this slide, I included the Novavax vaccine, which is in phase 3, but I also included 2 other protein subunit vaccines that might be available outside of the US before it's available in the US. All 3 of these vaccines are administered in 2 doses, 3 weeks apart. The advantages of these vaccines are that they have well-established production and distribution technologies. However, there's, because these are actually produced in culture, opposed to how we mentioned with the mRNA and adeno, that they make endogenous protein from, like, within our cells, we make the spike protein. These are actually produced in vitro. And there's, for instance, I guess the GSK Sanofi is made in baculovirus expression system, for instance. And so you always have to make sure that, like, the protein folding and the glycosylation, this kind of thing is still close enough to the native spike protein to make sure that the immune response is protective. Also, these vaccines have a slightly slower manufacturing process, which is, again, they're probably going to be third to the market in the United States. Next. So in this table, I just would like to briefly highlight some of the key vaccines in the world and some of their preliminary efficacy data. So in the first 2 rows, at a median of 2 months, both Pfizer and Moderna vaccines were 95% and 94% effective, respectively, in over 40,000 and 30,000 people. In the third row, the AstraZeneca vaccine, which is not yet authorized in the United States, but it's available in a number of nations, reported phase 3 efficacy of about 70% at 2 months. And in a subgroup analysis, they found that people who were given a half dose followed by a full dose achieved 90% of protection. However, patients given the standard dosing only had 62% efficacy. So AstraZeneca has now modified their phase 3 protocol to include additional arms to try to determine the optimal dosing. Again, J&J is expected to release phase 3 data as early as next week, and this could provide us with a very interesting piece of the adeno dosing puzzle. Novavax is also expected to report preliminary efficacy data in the next few weeks. Also in the last row, just for a comparison, I wanted to include some recent data from Brazil, which is the largest study to date of the Sinovac inactivated vaccine, which reported about 50% efficacy in 13,000 people. So note that only the Pfizer vaccine is, that's the only vaccine on this table that requires the ultra low temperature chain. So that's just something for people to note. Next slide, please. So in conclusion, the evolving landscape of COVID vaccines presents the global community with substantial complexity, uncertainty, and opportunity. It's important to continue to monitor the data in your respective options as things unfold in your nation. So the good news is that most vaccines are only going to require refrigeration between 2 and 8 degrees Celsius. And an important consideration is that each country will not have the same decision set in terms of your number of vaccine options and their safety and efficacy profile. However, the strong efforts by the global vaccine manufacturing community I'm very encouraged that hopefully very soon everyone in the world will have an acceptable and timely vaccine available. So I feel that informed clinical engineers can play a key role in helping their respective health systems best navigate this complexity in 2021 to optimize the outcomes in their nations. So thank you for your attention.
Great job, Marcus. Excellent coverage of something that we all have a question about. I appreciate that. And I know you don't play attorneys on television. There were a couple questions about the request for a waiver when you take the immunization. Abigail answered that. I wonder if you can, in the Q&A box, write something about why it's being requested. And Dr. Furman, if you can look at the question about ECRI involvement in UC Irvine in California research.